Hello, everyone, and welcome to Lunch Break Science. I'm Ariel Johnson from the Leakey Foundation, a nonprofit committed to funding human evolution research and sharing discoveries in programs like this one. Lunch Break Science is made possible through the generous support of Anna Gordon Getty and Camilla and George Smith. Here with us today is 2008 Leakey Foundation Baldwin Fellowship Scholar, Dr. Lauren Schroeder. We are so excited to have you here today, Lauren.
Thank you. It's wonderful to be here, and thank you, the Leakey Foundation, for this opportunity.
So Lauren originally hails from South Africa, but is joining us today from Toronto, where she is Assistant Professor of Anthropology at the University of Toronto. We'll be exploring her work, not only distinguishing the differences between the different features of skulls of our early ancestors, but also kind of where those differences originate from. So Lauren, my first question for you, why skulls? Why are skulls so interesting and important when we think about human evolution?
So I would say, firstly, skulls are cool, but more seriously, cranial and mandibular remains are quite well preserved in the fossil record. So even though fossils are, of course, very rare, with skulls we have a relatively largish sample size to work with.
Yeah.
Secondly, the skull has very species-specific morphological patterns, which have been commonly used in the past in investigations of variability. And then the third point is that the evolution of the genus Homo, which is the topic of my research, and the genus Homo is, of course, the genus which includes us, so Homo sapiens. This genus is characterized by a number of well-documented evolutionary trends in the skull, such as brain size increase, as well as shifting adaptations related to diet and chewing. So it has been at the core of many hypotheses relating to evolutionary change.
Yeah, I feel like there's something about a skull, because when you look at a skull with the eye sockets, you kind of have more of a connection, because we look at each other's faces. So I feel like it's kind of like looking at the face of our past. I don't know.
Absolutely.
Sorry. Before we get started, as a reminder to those of you watching us on YouTube, Facebook, and Twitter, you can leave questions or comments in the comments anytime for Lauren, and we'll get to as many of them at the end of the talk as possible. But if we don't get to your question, we will have Lauren be answering them through a blog post a little bit later. So we'll share that. Yeah, we're just so excited to have you here with us today. So, Lauren, how did you become interested in studying anthropology and specifically skulls? Like, what is that?
So, as a child, and I think this probably resonates with so many of you listening, I was really, really, really into dinosaurs. My parents bought me these weekly dinosaur magazines that each had like a piece of a puzzle skeleton of a T-Rex. So if you had all of these magazines, you could build your own T-Rex model skeleton. I guess a pretty good marketing gimmick. And together with my dinosaur obsession, I also had these books called How My Body Works, which I think got me into human anatomy. But then in high school, I moved a little bit away from these topics. I became very fascinated with astrophysics, and eventually I started my undergrad studies in the astrophysics program at the University of Cape Town. But by chance, I ended up taking a second-year course in human evolution with Professor Becky Ackerman, who would eventually become my mentor and advisor. And I think it was in the first or second lecture that I could say the human evolution bug bit. And I knew that at that point I just needed to change everything and pursue a career in paleoanth. So if I think about it, it's like I came full circle at that point back to my childhood obsession with anatomy and old fossils, not dinosaurs.
It's like it's really neat to get to do something, you know, that you wanted to do when you were a kid. I've heard a lot of scientists say that same thing, is that, you know, they did kind of return to that childhood curiosity, you know. And I don't know, it's such a great field. So, and we're really excited to have you here. Now, your work now is mainly based in the lab, and you're doing some amazing things. But early in your career, it was kind of more of a mix of lab and field, right? Do you want to talk a little bit about some of your opportunities there?
Yeah, sure. So during my PhD and earlier, I traveled to multiple institutions across the world to collect data on the most amazing fossil hominins, and also just as amazing skeletons of extant primates. So living chimps and gorillas and so forth. The pictures that are being shown include me with the town child on my first fossil data collection trip to the University of the Witwatersrand, which was about 13 years ago. The picture of me with all of those chimpanzee skulls was taken at the Cleveland Museum of Natural History. And then in terms of full field work, I was a student on the Kubi-Fora Field School in 2007, which really gave me I would say, first-hand experience of what it's like going to the field, the extreme hard work that excavation entails, and especially the fact that the Kubi-Fora region is so fossiliferous, and all of these famous fossil finds were from there. It really was quite a privilege to be able to I would say, to be a part of all of that. Recently, though, my collaborators and I are hoping to get back into the field. A lot of my work right now is focused in the lab. And we did an initial site visit to a site in the cradle of humankind in South Africa. So here I am with myself and, well, I'm with all my collaborators. So I'm definitely hoping to get back into the field soon, once COVID is done.
I know, right? It's so hard. There's so much, all this, you know, amazing, you know, research out there. But it's good that, you know, you can be in the lab and get some work done there, and, you know, participate in programs like this. You know.
Yeah, exactly.
You know, and you've had the opportunity to work with some very famous fossils. You've been involved in both the Malapa project with Australopithecus sediba and the Rising Star project with Homo naledi. You were actually, I believe, one of the team leads to describe the skull of Homo naledi. Do you want to talk a little bit about that?
Yeah, so the Rising Star workshop brought together multiple, mostly early career researchers, and we were tasked with conducting the initial analyses and descriptions of the fossils from the Rising Star cave. And I was part of the skull team, which included 5 other exceptional female scientists, Mana Dembo, Heather Garvin, Myra Lard, Davorka Ratkovich, and Jill Scott. And day after day, for 6 weeks, we meticulously described and researched the hundreds of Homo naledi cranial and mandibular fossil fragments. And I would say that the first time we walked into the vault at the Evolutionary Studies Institute at the University of the Witwatersrand, there was basically a sea of bones. Yeah. Many of the fossils were extremely fragmentary. So the first thing we had to do was to try to kind of fit some of the fragments together, like a really complex puzzle. And it really just initially takes your breath away when you handle these fossils of our ancestors. The picture that you showed previously is me looking at a 3D model of a rib cage that Dr. Marcus Pasteur was showing me. I had to put on 3D glasses. Yeah, and that's me with Australopithecus sediba. that I was also involved with in terms of the mandibular analyses.
So, when you look at fossil skulls, what are the major features and differences between them?
So on the screen, yeah, there you go. I've provided 3D models of two very famous fossil hominin crania. So on the left, you have Homo habilis, and as you can see, it's quite small. It has a very defined brow ridge, and then when you look at it from the side, you can see it has a prognatic face, meaning that the upper jaw kind of sticks out. The brain case is also rounded. We say it's quite high, and the back of the cranium is also quite rounded, but very wide at the bottom of the cranium. If we move on to the next specimen, which is an example of Homo erectus, you can see that it has a much larger brain size, very large brow ridges with a kind of gully behind them. The brain case is also quite long and low and kind of juts out at the bottom. And then when you look at the back of the cranium, you can see an almost heptagonal shape, so a seven-sided shape. And then this individual is still quite prognatic, but less so than habilis.
Well, now that all of you viewers have learned the differences between Homo habilis and Homo erectus, we'll actually give you an opportunity to try out your skull identification skills. So Lauren will share a mystery skull with us right now, and you will look at these 3 key identifying features that Lauren just taught us and assess what you think it is. Is it a Homo habilis, a Homo erectus, or possibly neither? I'm going to bring up the mystery skull now.
So I'll just go through some of the details of this mystery skull. So as you can see, the skull has a brain case on the smaller side, but it's quite round, which is similar to what we see in Homo habilis. But when you look at it from the back, it looks like it almost has a pentagonal shape, but also that it still has that kind of jutting out bit of bone at the bottom of the head. The brow ridges are defined, but not as large as what we see in erectus. And although the middle of the face is missing, we can reconstruct the prognatism here, so how far the upper jaw sticks out, and we could say it's quite prognatic.
So we'll give people a few more moments to respond in the comments. I think we have, we're starting to see some of the comments. Neither. What else do we have? Habilis. Oh. Any more comments? Oh. Maybe give a few more moments for guesses. Must be Homo habilis. So Lauren, what is the correct answer here?
So the answer is neither. This is actually the skull of Homo naledi. And I chose this mystery fossil to also just kind of show you how the Naledi skull team determined how similar and how different the specimen is compared to previously discovered species. And it was actually quite a complex task because, as you could tell, we found similarities and differences between it and erectus and habilis. So although I was part of the skull team, there were actually multiple teams working on different aspects or different skeletal regions of Homo naledi, and using all of the combined evidence collected from each of the skeletal regions, the teams then together came to the conclusion that the fossils found in the Rising Star Cave system were different to anything that's been found previously.
Well, this was a really fun activity. It's our first time trying out something like this, and I think it worked out pretty well. I hope you all enjoyed it. But identifying the differences between skulls is only, a little portion of what your research is. And I think probably, at least to me, the more interesting aspect of your research is really looking at the evolutionary processes that kind of, exhibit themselves in these differences. So I am now going to turn the floor over to you, Lauren, and give you a chance to talk a little bit more in depth on your research.
Cool. Let me just try to share this screen over here. Okay. There we go. So for this mini lecture, I'll be talking a little bit about my work on reconstructing evolutionary processes in human evolution, specifically within the genus Homo. So this is what my PhD focused on, and is work that myself and collaborators are currently expanding. So to start, let us consider that historically, human evolution was once viewed as a kind of linear process with ancestors and descendants connected in a way where one kind of evolved into the other. So you've all seen those kind of T-shirts, right? Back then, paleoanthropological research also focused mainly on the effects or products of evolution. So morphological descriptions and taxonomic classification. So in other words, which species is more closely related to the next, which are of course all important questions. However, as our knowledge of population variability has developed, this more simplistic narrative has been replaced by, I would say, a more sophisticated understanding of just how complex our evolutionary story is. And more specifically, recent fossil finds like Homo naledi that we just spoke about, as well as the development of new methods for studying the fossil record, has given us an interesting alternative to this kind of linear view of our evolution, with a scenario that used to look like this, or was depicted like this, moving towards something that looks more like this. So how has this new understanding helped us advance the field? Well, before, many studies were usually rooted in an assumption that the differences and similarities in hominin species were the sole result of natural selection. So specifically, how well these species are adapted to their environment. However, the genetic and morphological evidence that we have accumulated from studying other organisms, multiple other organisms, humans and non-humans, shows that adaptation via natural selection is not the sole process of evolution. It's just one of or one part of a combination of both adaptive and non-adaptive evolutionary processes. So in other words, evolution, or the change in inherited traits of a population through time, also includes an accumulation of chance changes in a population's gene pool through time, which is called genetic drift, as well as hybridization between different populations, which results in gene flow. But the question is, how do we study these processes in the fossil past? Of course, this can be a bit tricky, given that when we go very far back in time, we don't have genetic data to work with. So sure, we can get some ancient DNA from Neanderthals and some of our other more recent relatives, which has, for example, provided us with tangible evidence of interbreeding and gene flow between populations. But when we go further back in time, this is more difficult. Luckily, though, there have been theoretical advances in the field of evolutionary quantitative genetics that allows us to estimate and model how morphology will act under certain scenarios. So, for example, the effect of genetic drift on morphology versus the effect of natural selection on morphology. So I just thought I'd quickly kind of graphically explain what I mean. So simply, if you have two populations and they diverged under genetic drift or random chance, we expect that the variability between those populations will be proportional to the variability within each group. So this is seen depicted on the graph on the left. On the other hand, a non-proportional relationship would indicate that between group variability, between these groups that you are studying, is more or less than expected. And this suggests that natural selection may have played a more important role. So, using this approach in the deep fossil past, my focus for a big part of my career was to explore the evolutionary processes that produce the patterns of variability seen during the evolution and diversification of our genus Homo. And I did this by trying to test the extent to which adaptive versus non-adaptive explanations were, which ones of those were more supported. And then the eventual aim was to explore possible correlations between my results and also common adaptive hypotheses within our genus. So common ones like diet changes and brain size increases. So 3 interesting results emerged. The first is that when I looked across the genus Homo, more comparisons than expected were not indicating strong evidence of selection. Rather, they indicated a more complicated pattern that included non-adaptive processes. Surprisingly, this pattern was also seen in the brain case, which kind of questions the traditional view that selection was the main driver of brain case shape within Homo. Now, this doesn't mean that having a large brain didn't ultimately provide benefits. Of course, it did. But what it is saying is that it is fairly easy to evolve a Homo-like brain or a Homo-like brain case through chance changes or chance effects, because the patterns of variation that I spoke about a little bit earlier, if you look at those patterns across populations, they are quite consistent through time. And then finally, I did see a very strong adaptive signature in the jaw, which does support previous hypotheses of jaw diversity and jaw diversification, and may definitely relate to dietary shifts and chewing adaptations, because we know that smaller jaws were kind of selected for during our evolution of the genus Homo. So to end, what does this all mean, and what are the implications? So in this mini lecture, I hope that I kind of provided all of you with a clear overview of just how complex the evolution of our genus has been. It has been a mixture of multiple evolutionary processes, and also, as this image on the slide shows, it's also been a mixture coming together and splitting up of populations, and all of this eventually giving rise to the diversity that is us today. So that's it for my mini lecture. Thank you. Oops.
Thank you so much. That was an amazing talk. Please, everybody, ask your questions in the comments if you're watching live, and we will be pulling up some questions shortly. Do we have any? So some scientists doubt the existence of Homo habilis. What are your thoughts, Lauren?
Oh, that's a tricky one. Species species identification in the fossil record is always difficult. In the 60s, when Homo habilis was first, would say, announced, there were lots of debates surrounding whether, as this, I guess Eunice says, there were lots of debates about whether it was a valid species or not. These days, I would say that it is recognized as a valid species, given that a lot more fossil evidence of Homo habilis has been discovered that kind of fits into what we would call a paleo species. Species concepts are also really difficult and complicated when we're looking at the fossil record. But I would say it fits within a paleo species definition, definitely.
Here we have one from Cheryl Camisa. She is our executive director of the Leakey Foundation. So thank you so much for the question, Cheryl. If you have time and money were no issue, what is the one question of human evolution that you'd like to answer?
Time and money. Well, for sure, I would try to figure out the most likely common ancestor or ancestral group of the genus Homo. We have this kind of scattered evidence from South Africa and East Africa, and it would be really cool to excavate some more sites and find more fossils in both of these regions. To kind of answer that question for sure. I mean, if I would say if I could go back in time, the question I would really want to know is, or what I would really want to do is probably collect DNA from all of these ancient fossils, if I could.
Yeah, you have to have them sign waivers too, I guess. So what would be your advice to aspiring paleontologists? How do they get started? This is a very good question.
Yeah. Thanks, Rebecca. I would probably say go to a field school, attend a field school. You'll either love it or hate it.
We'll know at least. We'll
know. No, for sure. I mean, the field school opportunities that I had as an undergrad and then at the beginning of my grad school career really solidified my interest in the subject. Also, doing a lot of reading, finding gaps in the literature was definitely something that helped me as a coming up in my career. But field school opportunities and field school, just attending and going to a field school is really something that I would suggest.
So I think we've got time for one last question. What is the oldest Homo or related fossil that yielded DNA? It's okay if you don't know the answer to this question too.
So currently we have, I would say the oldest biomolecule that has been yielded from fossil Homo was from Homo erectus in Dmanisi. This is not ancient DNA, but ancient proteins. So we have ancient proteins from Homo erectus, and then we have ancient DNA from Neanderthals and also the Atapuerca hominins, so more Middle Pleistocene Homo from Spain. Yeah.
Wonderful. Well, that's a good question. It was supposed to be our last question, but maybe we'll take one more. Well, what does the future of your research look like, and where to next?
Thank you, Jenna. Jenna is actually my my very good friend. She took all of those pictures of me incubator. So wonderful. So currently, my work is still focused on evolutionary process, but I've expanded to hybridization. So I'm not going to have time to talk much about this right now.
We'll have you back just to talk about that, because it's really interesting research too.
Yeah. Currently, just a little tidbit. Some of my students are working on a project looking at hybridization in coyotes. So from ancient hominins to canids, but they're trying to figure out the morphological signature of hybridization in living animals, and then trying to model what that hybridization will look like in the fossil record. So that's where I'm off to next.
Well, thank you so much for joining us, everybody who's viewing, and thank you so much, Lauren.
Visit us at leakeyfoundation.org to learn more about the Leakey Foundation, Lauren Schroeder, and how you can help support research like Lauren's and educational programs like Lunch Break Science. Right now, all donations will be doubled by 2 generous donors, meaning your impact will be doubled. Next Thursday, we meet Leakey Foundation scientist Chalachu Sayum and hear about his work reconstructing the diets and environments of our ancestors. Be sure to subscribe to the Leakey Foundation's YouTube channel, follow us on Facebook and Twitter, or sign up for our email newsletter to be the first to hear about upcoming episodes, groundbreaking discoveries in human evolution, and much, much more. Still hungry for science and can't wait until next week? Check out the Leakey Foundation's award-winning podcast, Origin Stories, available wherever you listen to podcasts. Lunch Break Science is made possible by the generous support of Anna Gordon Getty and Camilla and George Smith.
Well, thank you all for taking a break from your day and feeding your brain with the Leakey Foundation. Until next week, stay hungry for science. Bye, everyone.
Bye.